Students, we were learning chapter biomolecules. We will continue that chapter biomolecules. So, students, in our last class we have discussed about the structure of proteins, isn't it? Structure of proteins we have discussed in our last class. If we have to do a recap what we have learnt in last class, we have learnt the structure of proteins, that is, proteins exist in 4 levels. There are 4 different structures. That is, first one is primary structure, which is like a straight line where one side it is called N terminal and other side it is called C terminal. No? So here each amino acid can be seen, which is the first amino acid, which is last amino acid. Easily you can see. Sometimes protein exists in a helical way or like a beta plated sheets, isn't it? So this is alpha helix and this is beta plated or B plated sheet. This is nothing but secondary structure. Then we have seen that it is slightly coiled, that is tertiary structure. Then if they are coiled, highly coiled, then that is quaternary structure. Now, to have particular biological activities in the body, it should have some coiled shape also. So this is what we have learnt in our last class. Today we will learn about what kind of bonds are there between biomolecules. Which type of bonds are there, like nature of bonding, between monomer, between monomers in a polymer, or linking monomers in a polymer. We can make it very simple. Nature of bondings, linking monomers in a polymer. So meaning is very simple, like what we have seen. Suppose protein is a polymer. Why it is called polymer, you know? Because protein is a long chain of amino acids where each amino acid is a monomer. Like we'll take example, like this is one amino acid 1, no, first amino acid joins to second amino acid. I'll write amino acid as AA2. Then one more amino acid binds, this is AA3. One more amino acid binds, then it is AA4. Like that it continues. No, it continues. So each amino acid is called monomer. Mono means single unit. Like this is like saying, to make one wall, to build one wall, many stones are required, isn't it? Each stone is a monomer, single unit. When many single units join, it becomes a many units, that is called polymer. So each amino acid is a monomer. One monomer, second monomer, third monomer, fourth monomer. Like that many one units join and become many units. When it becomes many units, then it is called a polymer. So what kind of bonds are there between these monomers in the polymer, we have to see. Okay, what is this bond? If it is a protein, protein is a polymer. Okay, it has bond called a peptide bond, you know. So nature of bond in a protein is peptide bond. How this peptide bond is produced between two amino acids? Peptide bond is produced only when carboxyl group, COOH group of one amino acid binds to amino group of other amino acid. Meaning, see here, this is one amino acid, this is other amino acid. COOH group of this amino acid binds to amino group of this amino acid. That is how bond is produced. So in a protein, between two amino acids, bond is produced because of carboxyl group of one amino acid binds to amino group of other amino acid. Then only it can form a bond, and nature of bond formed is peptide bond. Then what about sugars? Like polysaccharides we have learned, which is a carbohydrate which is produced by joining of many sugars, like glucose, glucose, glucose many times, or fructose, fructose, fructose many times. If fructose, fructose, fructose joins and becomes a long chain, it is called inulin. Already you have learnt it, no? So suppose if you take the example of polysaccharides, which is a polymer of sugars, many sugars join and becomes a polymer, and the nature of bond formed is glycosidic bond. Okay, individual monosaccharides, like this is suppose glucose, okay, then joins to one more glucose, joins to one more glucose, joins to one more glucose. This is a homopolymer where same monomers are repeated. Suppose this is glucose, then there is fructose, like that different amino acids join, okay, then it is a heteropolymer. Whatever the polymer is, between two sugar units, the bond formed is called glycosidic bond. Glycosidic bond. Always between two sugars, like between two glucose, the bond formed is called glycosidic bond. Okay, so this is formed by, glycosidic bond is formed by dehydration, that is removal of water. D means removal, hydration, hydra means water there. So by removal of water molecule, glycosidic bond will be produced. Okay, so bond is formed between two carbon atoms of adjacent monosaccharides. Bond is formed between two carbon atoms of adjacent monosaccharides, like carbon atom of one monosaccharide with carbon atom of other monosaccharide, so that bond is produced. That will be called as a glycosidic bond. Okay, in case of nucleic acid, that is DNA has a slightly different structure. If we have to see DNA structure, where DNA is a structure like this, helical structure like this. Okay, this is structure of DNA, that is deoxyribonucleic acid. You can write N capital. So it is D N A. Already you know. Okay, I don't have to tell you where there are two strands here. Two strands means two ribbon-like structures coiled like this. Each one is called a strand, no? Strand. So two strands are there. Strand means thread-like structure. Okay, and both the strands will run anti-parallelly. Run anti-parallelly means like one strand is having 5' to 3' end, then other one will have 3' to 5' direction. What is that 3', 5'? This is 5' means phosphate group is attached to 5th carbon atom of sugar. You know this already, you have learned what is nucleotide, no? Already you know what is nucleotide. Nucleotide means it will have 3 parts, like phosphate group, and phosphate group is attached to pentose sugar, attached to 5th carbon. So this is oxygen is there here, then this is carbon 1, this is carbon 2, this is carbon 3, carbon 4, and this is carbon 5. See, phosphate group is attached to 5th carbon atom, so that becomes 5' end. Then here this will join where this pentose sugar joins to nitrogen base, adenine, guanine, thymine, cytosine, like that. Okay, so this is nucleotide. Nucleotide will have 3 parts: phosphate, pentose sugar, nitrogen base. Already together all 3 are called nucleotides. Nucleotide. So DNA, if you see the backbone of this DNA, if I zoom this thread-like structure, inside the thread-like structure there will be 3 parts: phosphate, pentose sugar, nitrogen base. Where in each thread, on each thread, if I zoom one small part which looks like a thread like this, but that thread-like structure will have phosphate, and to that pentose sugar, and to that nitrogen base like A, T, G, C will be attached. So this is a nucleotide. On each strand, here also nucleotide, here also nucleotide will be there. Okay, so 5' end means where phosphate group is attached to 5th carbon atom, that's why it is called 5' end. 5 means 5th carbon atom. And this is called 3' because OH group, hydroxyl group, is attached to 3rd carbon atom of end of that strand. Like you start with this strand where phosphate is attached to 5th carbon atom, then at the end of this strand, OH group will be attached to 3rd carbon atom. So that's why we say one thread, like one strand of DNA is having 5' to 3' direction, and other strand will be having opposite direction, that is 5' to 3' direction. This is what we call, they run anti-parallelly. Anti-parallel, like one strand goes from 5' to 3', other one will go from 3' to 5'. 5' to 3', 3' to 5'. So they will be having anti-parallel. If both runs together, it is parallel. If it is running in opposite direction, then it is anti-parallel. You see there. Okay, that is what they are talking about in a DNA. Okay, then very important thing is what? The bond formed between phosphate and sugar is what? Bond formed between phosphate and pentose sugar. What is this bond? Here color chalks are not there, otherwise I would have highlighted. Bond in a nucleotide, bond formed between phosphate and pentose sugar is called what? It is called ester bond. Ester bond. Bond formed between phosphate and pentose sugar in a DNA where nucleotide is there, it is called ester bond. Okay, ester bond. And this is about one strand, means one thread like structure. One more will be there here. Here also phosphate comes, here also phosphate comes, here also pentose sugar, here also pentose sugar. So here one ester bond and here one ester bond. If ester bond is there twice, then it is called phosphodiester bond. Phosphodiester bond. Why phosphodiester? Because phosphate group, phospho means phosphate group is attached to pentose sugar with the help of ester bond. Then it became phosphate plus ester, phosphoester. And as this bond is present on both the strands, here also, okay, that's why it becomes 2 ester bonds, 2 phosphoester bonds. Then it is called phosphodiester bond. Di means 2. Okay, so phosphodiester bond. Bond formed between phosphate and pentose sugar is ester bond in a nucleotide, and nucleotide is present on the DNA. Okay, so who gave double-stranded DNA structure of, who gave this structure of DNA? By 2 scientists. Their name is Watson and Crick. That is what is given in the textbook. Their full name is James Watson and Francis Crick. two scientists. They gave structure of DNA. Okay, so in 1953, James Watson and Francis Crick. Okay, in short, they are called Watson and Crick. They explained, okay, they discovered structure of DNA, how it looks like, they explained, and characters of DNA also they explained. They said it looks like a double-stranded helical structure like this. Okay, so see, helical structure like this. So you have to draw in this way only. See, this is wrong diagram. If you draw exactly like this, this is wrong. Okay, where gap is very less, it comes close. See how you should draw, I'll tell you. DNA structure while drawing, first randomly you draw, but while bringing, you should not make it broad like this. You should bring it close. See, close. Like this it should go. This is the correct structure of DNA. Okay, so this structure of DNA, where one strand is 5' to 3', other one is having 3' to 5'. I told you what is the meaning of this. 5' means phosphate group is attached to 5th carbon atom of pentose sugar, and here OH, hydroxyl group is attached to 3rd carbon atom of pentose sugar. Okay, so this is a double-stranded DNA structure given by Watson and Crick. It is famous model known as double-stranded helix model. Double-stranded helix model of DNA. What is meaning of helix? Two thread-like structures, two strands coiled like this. That coiling is called helix. You know, like a staircase, helix. Okay, then the backbone of this, if you see what is there, if you zoom it, what is there in each thread, I told you there are nucleotides. Nucleotides are there, like here phosphate is there, on this strand also phosphate is there. Then here pentose sugar is there, here also pentose sugar is there. Then nitrogen bases will come here. Like suppose A is there, then on opposite side T comes. I'll tell you why. It is because you know that already A and G, adenine and guanine, are purines, one type of nitrogenous compound. Then cytosine, thymine and uracil is called pyrimidines or pyrimidines, one type of nitrogenous compound. Already this is 3rd or 4th time we are discussing this. Okay, a scientist called Erwin Chargaff, his name is not there in the book, he has given a rule of complementarity. That means on one strand of DNA, suppose A comes, okay, always opposite to that T comes, or vice versa, nothing else will come. Okay, and A and T will have two hydrogen bonds, two bonds or two hydrogen bonds will be there. And when G comes on one strand, on one thread, and on opposite side there will be C or vice versa, and it will have 3 hydrogen bonds, triple bond. This is double bond, this is triple bond. So 3 hydrogen bonds. Vice versa means what? A can bind with T or T can bind with A, not with any other things. Or G will bind with C or C will bind with G. Why? Because he has given a rule that is called A plus G is equal to T plus C, where A binds with T, G binds with C, or vice versa. I'll tell you why he said like that. Because he said in a DNA, number of purines are always equal to number of pyrimidines. Means how many purines will be there, that many pyrimidines will be there. Equal number. Like if A is there, then T will be there. Or if T is there, A will be there. Like 50 A's are there, then 50 T's will be there. So he says purines always come opposite to pyrimidines, and number of purines is equal to number of pyrimidines. This is the rule, but this much explanation is not there in the book. I just told you because next year anyway you are going to learn about this again, especially about DNA. So A binds to T with 2 hydrogen bonds, double bond. G binds to C with triple bond. Okay, always. That is hydrogen bonding will be there. Hydrogen bonds. Okay, that is what they are saying. Here, suppose here A is there, then opposite to that T comes and they will have double bond. Okay, if G is there, then opposite to that C comes. If C is there, opposite to that G comes. Okay, if T is there, opposite to that A comes. Okay, this is how bonding occurs. That is also called complementarity. Complementarity means if A is complementary to T, T is complementary to A. Okay, but A and T are not complementary to G and C. G is complementary to C, C is complementary to G. That means what binds to what, that they have explained. Okay, so what is the backbone? Backbone of DNA is phosphate, binds to pentose sugar, then nitrogen base. So phosphate, pentose sugar, nitrogen base is a nucleotide, you know already. Once again, let us recall what we have learnt in last class. If phosphate group binds to pentose sugar, then it is called, what it is called? Sorry, what I was saying, sugar. Where nitrogen base like adenine binds to sugar, then it is called nucleoside. For example, adenine plus sugar, it becomes nucleoside. Plus phosphate, if 3 are there, if phosphate also joins, then it becomes a nucleotide. No? So nucleotide is a backbone of